This conference will now be recorded.
Hello students, I'm Dr. Divya from TeenEinstein. So now we are in the 10th chapter, microbes in human welfare. So this is our second class. So microbes in human welfare part 2. In the first class, that means part 1, we were discussing about the role of microbes and overall introduction we gave. Then we have discussed about the presence of microorganisms. Actually, they are present everywhere. Like they are present in the soil, air, water, and some of the microorganisms, they are present in our body also. So in animals, then and plants and all, everywhere they are present. So wherever the normal life is impossible, like in very hot condition or in severe cold condition, in all such type of conditions also some microorganisms are living. So for example, in extreme hot condition, in extreme cold condition and under soil, so in such different types of microorganisms are there where they can live under severe conditions also they can live. So such type of microorganisms are there. Then you know about different types of microorganisms. They are bacteria, fungi, protozoa, then plant viruses, animal viruses and all. These are different types of microorganisms. Then some more types of organism we had learned in 11th standard. Hope you remember. This we didn't discuss in the last class. So they are viroids and prions. We have learned in 11th standard. For NEET exam, actually this is important. Okay, so in the last class, hope you don't have any other doubts. So we are moving to part 2, second class. So first we will discuss about the viroids and prions. So let us start with the general points of viroids. Okay, you have already learned this in 11th standard. So there's the main points we are discussing here because it may be useful for your NEET exam. So viroids, mainly these viroids, they are pathogens. Okay, viroids are mainly pathogens and they affect plants. They affect the plant body. The pathogen means they may cause some disease. So they will affect plants only. Okay, plants. So they are mainly plant pathogens and especially in higher plants. Okay, not all type of plants, especially they are in higher plants. Note down all points because only important points I have taken. So the viroids, they are plant pathogens and mainly they are affecting higher plants. Okay. So we can call them as plant pathogens, viroids, because they are affecting mainly plants and that also higher plants. So we can call as plant pathogens also. Then these viroids, viroids are smaller than what? Smaller than viruses. Okay. They are smaller than viruses. So viroids are smaller than viruses. Then these viroids, you can see this is the structure compared to and they have RNA. RNA means ribonucleic acid and which are as circular strands. You can see this as circular strands and that these strands here you can see this is the type. These are the viroids. Here you can see they have protein. One second. See, for viruses, how it will be, the genetic material, it is covered with protein, right? But in the case of viroids, here you can see they have the genetic material, but it is not covered with any protein coat. So there is no protein coat. No protein. They don't have protein coat. So what and all we have learned? Viroids. Viroids means they are pathogens, mainly they are plant pathogens, and in higher plants we can see. Okay, mainly they are affecting the higher plants, and you know they are smaller than viruses. Okay, when they are asking about viroids, you have to write all these points. These are the main points. They are the plant pathogens, especially higher plant pathogens, and they are comparatively smaller than viruses, and they don't have protein coat. We said, see, viroids, now we are comparing with the virus. So when they are asking to compare, it is smaller than virus, then they don't have protein coat. In the case of virus, they have genetic material which is covered with protein coating. Protein coating. Okay. Then, when this viroid it is entering in a plant, what it will do? This, see, here you can see that is having only a genetic material. So when it is entering, this is a plant cell. When it is entering the plant cell, what it will do? That plant, it is entering or it is utilizing the complete system of that plant. For what? Then only it can replicate and that can survive. So what it will do? When it is entering inside the plant, it is utilizing that system, that plant cell system. Completely it is utilizing that plant system. Okay. So this viroid, that genetic material is getting dominated there, or that is going to control the whole process. So when it is entering inside, that utilizing the complete system of that plant for replication. So here it is getting replicated, and that's how it is getting that survivor. Okay. So whatever system in that plant, it is present, whatever it may be, whatever is present, whatever it may be, everything, all systems will be used by this viroids. Okay. So when it is entering inside the plant cell, then the overall control of that plant cell is taken by this viroids, and that's how it is getting replicated and surviving. Okay. So everything is being used by this viroids. So here in this picture, you can see the structure of that viroids. See, this is the structure of viroid. Actually, you can see that they don't have a structure like virus. Just now I said they don't have a structure like virus. And they have a single-stranded RNA without that protein coat. Okay. For virus, how it will be? The RNA or the genetic material with protein coat you can see in a virus. But here they don't have that protein coat. They have single-stranded RNA. Okay. Single-stranded RNA, then no protein coat. Protein coat. Without protein coat, they are. Okay. So they don't have that protein coat, and they have single-stranded RNA, then without that protein coat, and they have, here you can see that short strand of circular DNA. They have the circular DNA, which is circular RNA. Okay, they have small strands of RNA. They don't have long strands. That circular, it's covered. Okay, it's not open. That covered RNA, it means closed system. This is a closed RNA which is having short strand of circular RNA. Okay, So when the plants are getting this infection, I said it is affecting the higher plants, right? First, it is affecting the higher plants. So when the plants are getting infected with these viroids, that may cause the complete loss of that crop. Okay, that whole crop may get destroyed, or that will get affected completely. They will get affected. So for that affected crops, examples are, means normally in this kind of crops you will find the infection. Coconut palms, then cucumber, then tomatoes, then potato, then avocado. Okay, so what are the pathogens? They are the pathogens, viroids, they are the pathogens, and they are affecting plants. Mainly they are affecting the higher plants. Then you can see it is smaller than the viruses. Okay, and it is smaller than viruses. Why we are saying like that? And that is a closed genetic material in that. Okay, single stranded RNA which is closed, and there is no protein caught around that. Here you can see there is no protein caught around that. And examples, which are all the examples: cucumber, tomato, potato, avocado, coconut palm. Normally in these plants you can see this kind of infections. Okay, then we said these viroids, they have RNA. It is having RNA. Viroids have RNA, and that is infecting only plants, that we have told. So a single stranded RNA. Here you can see it's single stranded, not double stranded. So the RNA which is single stranded. The RNA is single stranded, and that is having what? Low molecular weight. Low molecular weight. Okay, low molecular weight. So RNA is single stranded with low molecular weight. Okay, and they'll be having unique structure. You can see here they are having that RNA, particular RNA is having unique structure. And what these viroids will do, they will replicate within that host cell. See, when it is entering into the plant cell, it will get replicated within that host cell, and that affects completely that host cell. And finally, what will happen? That host cell, that will lead to the death of that host cell. Okay. So what is happening? That RNA, the genetic material, or it is having only RNA, viroids, and that infects only plants, that we have already told. So this RNA which is present, it is single stranded with particular structure, or they will be they are single stranded with a special structure. You can see here. And that RNA with a single stranded, that is having very low molecular weight. Okay, that RNA will be having very low molecular weight, and they have special structure or unique structure they are having. And viroids, what they will do, they will enter inside the host cell. See, the viroids will enter inside the host cell, and inside the host cell it will get replicated. Then that is affecting that host cell. Then finally that will lead to the death of that host cell also. Okay, finally it is destroying or disturbing that host cell. Okay, clear? Then next, now we have learned about which one? Viroids. So next important one is another microbe, that is prions. Okay, prions. So what are these prions? Prions, we said they are proteinaceous infectious particles. Okay. Proteinaceous infectious particles. They are the proteinaceous infectious particles, and these prions can cause so many diseases in mammals. Okay. Like the diseases like neurodegenerative diseases. For the neural system and all, it is causing diseases, neurodegenerative diseases. For example, we can say that Creutzfeldt-Jakob disease. Okay, that is in human. In human, you can see Creutzfeldt-Jakob disease. Okay, Creutzfeldt-Jakob disease. That is a human being. In human, you can see this disease. Okay, so prions means, what is prions? They are proteinaceous infectious particles. Okay, that with protein, it is made up of proteinaceous infectious particle, which is causing infection. So they can cause so many diseases in mammals. Especially in mammals, it is causing many diseases like neurodegenerative diseases. They are causing neurodegenerative diseases. They are mainly causing neurodegenerative diseases. Okay. See, some kind of protein folding. Here you can see the protein is folded. So, in this, or the prion, when it is getting infected, the protein folding, how it will be? That will be abnormal folding. That's why this disease is coming. From the normal protein, from normal protein folding, it is getting folded in an abnormal way. Okay. So from the normal protein folding, it is getting folded in an abnormal, or abnormal protein folding is found in this kind of diseases. Actually, this prion transmission, it is difficult. It's not so easy compared to other disease transmission. Prion transmission is difficult. So from one person to another person, the disease transmission is difficult. Okay. But the important thing is that disease transmission is difficult. But whatever may be, when it is coming, they are the neurodegenerative diseases. They are infecting the neurons. So neurodegenerative diseases, and they don't have a treatment at all, or they don't have a treatment or a cure. Okay, we said that is neurodegenerative diseases. So when the it is a neurodegenerative disease, when it is not getting treated, what will be the condition of that person, or what will be the condition of that infected one? So that will completely, the nervous system or the neuro system, that will get affected, right? So this is not only present in human being, but this is, you can see this is in cow and sheep and all such. Cow, sheep and all also you can see. In cow, the mad cow disease, this we have learned. Mad cow disease. Okay, this we have learned in 11th standard. Then mad cow disease is reported in cows. Then prions, they mainly causes this folding, abnormal folding of proteins, we said. So that causes the abnormal folding in the brain. Okay, and that is resulting in brain damage also. So finally, what will happen if abnormal folding is found in brain? Brain will get damaged. Okay, so viroids. Then another one is prions. In prions, what all we said? Prions means they are proteinaceous infectious particle. Okay, proteinaceous infectious particle. It is made up of protein or folded protein. So they can cause several diseases in mammals. Like all the diseases are neurodegenerative diseases. Okay, Creutzfeldt-Jakob disease. That is in human being. So what will happen? The abnormal folding of protein. The protein is getting folded in an abnormal way. So the disease is coming because of that. Actually, the transmission from one person to another person or one organism to another organism. This disease, which disease? This prion. Actually, that prion transmission is difficult. Okay, so the spreading of disease is difficult from one person to another person or one organism to another organism. But it is affecting the neuro system. So it is a neurodegenerative disease. The neurodegeneration is taking place. So they don't have a particular treatment or proper treatment or a care. Okay, the neural system neural system it is getting damaged. It is getting degenerated. So there is no treatment or cure for that particular disease. So not only in human, but in sheep and cow also, this kind of problems are seen or this kind of diseases are reported. So prion, it mainly causes abnormal folding. Okay, the protein is getting folded in an abnormal way. So in the brain and it is causing damage. Clear? Then, now prions and viroids we have learned. Next. So those portions are important for NEET exam, especially it is for NEET exam. Actually, it is there in 11th standard. Next, we are going to learn about role of microbes in industrial products. See, microbes, they have very good role in industrial products for the synthesis of industrial products. So many products are getting synthesized with the help of this microbes or microorganisms are used. So many microorganisms are used for the production of different products. Okay, and all these products, they are valuable and they are useful, very useful for humans. And you know about antibiotics, then beverages and all, right? So for the large scale production, the beverages and antibiotics or such things and all, for the large scale production of such things, for beverages production and all, or industrial scale production, for the production in an industrial scale. Industrial scale production, what we need? They need more microbes. Okay. So what they will do if they need more microbes? The microorganisms will be grown in very large vessels. Okay. For the industrial scale production of some products, the microorganism will be grown in very large vessels. So those vessels are called as fermenters. Microorganism can be grown in large vessels. So they are called as, these large vessels are called as fermenters. Okay. So what are fermenters? The vessels which are used for the growth of microbes for industrial scale production of different products. Okay. Here you can see in the picture. You can see in the picture fermenters. So what are fermenters? These are the fermenters. What is that? The vessels, big vessels which are used for growing the microbes for the industrial scale production of different products. Okay, here you can see in this picture the fermenters. Then this is the one more question. Fermenters, they are the vessels which are used for growing the microbes for the industrial scale production of products. Okay, and here the picture is also there. Now let's see about some industrial products in which microbes are used for the synthesis. So for synthesis purpose, in which and all industrial products we are using microbes. So let's see one by one. So first, for the production of beverages, microbes are used. You know that. For beverages production also microbes are used. Okay, so let's discuss about that first. What is the role of microbes in fermented beverages? Okay, you know yeast. Yeast. You know that. So yeast is a very, very important microbe and that is used for the production of beverages. Beverages like which and all? Wine, then brandy, then beer. Okay. So yeast is used for the production of beverages like brandy or rum or wine. Then beer, wine, brandy or rum and all. These things are produced with the help of yeast. That is the microorganism role of yeast in the production. Then that same yeast, what is the scientific name of yeast? Scientific name of yeast? Saccharomyces cerevisiae. Okay, Saccharomyces cerevisiae. So this is the scientific name of yeast, Saccharomyces cerevisiae. It is used for bread making also. Bread making. Okay, Saccharomyces cerevisiae is in bread making. It is having very good role in bread making. Then this yeast or the Saccharomyces cerevisiae, it is also called as brewer's yeast. Okay, it is used in the production of, don't forget, yeast. What is the scientific name of yeast? Saccharomyces cerevisiae. It is in bread making, then wine, brandy, beer and all. This is used and it is also called as brewer's yeast. Okay. Brewer's yeast. Okay. Saccharomyces cerevisiae, it is also called as brewer's yeast or baker's yeast. baker's yeast. Okay, it is either called as brewer's yeast or baker's yeast. Which one? Saccharomyces cerevisiae or yeast. It is called as brewer's yeast or baker's yeast because it is used in brewery and bakery also, right? So the yeast or Saccharomyces cerevisiae is also called as brewer's yeast, and it is used in the production of ethanol. Okay, this is used in the production of ethanol. It is used in the production of ethanol. Okay. So this malted cereals or fruit juices, what they will use? Malted fruit cereals or fruit juices, they are fermented with the help of this yeast. Okay, or Saccharomyces cerevisiae or yeast is used for the fermentation of this malted cereals or fruit juices. Then what it will do? And from that they will produce this wine, brandy, beer, ethanol and all. Okay. So in the same thing, the same yeast, this yeast or Saccharomyces cerevisiae, it is used in bread making also. And it is called as baker's yeast. So the Saccharomyces cerevisiae is also called as brewer's yeast and baker's yeast also. Then this malted cereals and fruit juices, they are fermented by using Saccharomyces cerevisiae to produce what? To produce ethanol. Ethanol is getting produced. Here we mentioned that ethanol is getting produced by the fermentation of this malted cereals or fruit juices with the help of this yeast or Saccharomyces cerevisiae, and different types of alcoholic drinks are produced. Okay. When it is getting fermented with the help of what? Saccharomyces cerevisiae or yeast, and it is producing which one? It is producing ethanol. Then these kind of alcoholic drinks are produced with the help of the Saccharomyces cerevisiae, and the production is based on the raw material. Which product is forming? That is based on the raw material which is using. Okay. So which raw material is using for the fermentation, and based on that the product will be there. Okay. Based on that the final product, the end product will be there. So alcoholic drink. Alcoholic drink. Alcoholic drinks, they are based on the raw materials and type of process which is going on. Okay. So there are types of alcoholic drinks. Based on what they are forming different types, based on which raw material is used and which type of process is going on. Based on that, the alcoholic drink will be formed. Then alcoholic drinks, you know that with distillation, which are all getting produced with distillation. Distilled ones. Which are the distilled ones? Whiskey, brandy, rum and all. These are coming under distillation. And without distillation, wine and beer. Wine and beer distill and without distillation. Sorry. Here, two types are there. First one is distilled and Without distillation. Okay. Without distillation is wine and beer. Okay. With distillation, whiskey, brandy. Whiskey, brandy, rum and all coming under distillation, distilled ones. So without distillation, wine, beer and all. So these products which are produced with the help of distillation or with the distillation of this fermented broth, it is called as, they are called as the distilled one. After distillation, it is getting produced. So the products are produced by the distillation of that fermented broth. By distillation of fermented broth, you may get one mark question. Fermented broth distilled ones. Which are they? They are rum, whiskey, brandy and all examples for that. The fermented broth is getting distilled. Then the, such kind of processes are going on in the production of whiskey, brandy, rum and all. But without distillation, wine, beer and all. Okay, wine, beer and all without distillation. With distillation of that fermented broth, rum, whiskey, brandy. Examples you can note down. Then here you can see the fermenters. In this picture you can see the fermenters and fermentation plant. This is the fermentation plant, the whole plant. And inside this plant you can see this kind of fermenters also. Okay. So what and all we have learned? Industrial scale production with the help of microbes. Okay. So first we are learning about fermented beverages. Right. So in large vessels, the productions are going on. In industrial scale, production will be done. So that large vessels are called as fermenters. Okay. Then Saccharomyces cerevisiae or yeast, that is very important microbe and that is used for the production of beverages like brandy, rum, wine, beer and all. Okay. So the same thing, Saccharomyces cerevisiae, it is otherwise called as yeast. The scientific name is Saccharomyces cerevisiae and that is used in bread making also. The same thing is used in bread making. The yeast or Saccharomyces cerevisiae, it is also called as brewer's yeast and it is used in the production of ethanol. Okay, so it is in brewery, it is used, so it is called as brewer's yeast. It is used in the production of ethanol. The fruit juices or the malted cereals, all these are fermented with the help of Saccharomyces cerevisiae or yeast. Then that same yeast is used in the bread making also. This Saccharomyces cerevisiae or yeast, it is used in the bread making also and that is this yeast, it is commonly called as brewer's yeast. Here, the Saccharomyces cerevisiae, it is also called as brewer's yeast or baker's yeast because in both cases it is used. Then, these malted cereals or these fruit juices, they are fermented by Saccharomyces cerevisiae and after that they will produce ethanol. After that fermentation, it will form ethanol. Okay. Like this, different types of alcoholic drinks are produced. And the production, it is based on what? The production is based on the raw materials. The raw materials which are used for that fermentation process. Based on that only the product. Okay. So based on the raw material, alcoholic drinks with based on the raw material which is used and the type of process which is used. So based on this, there are type of alcoholic drinks. Okay. Then alcoholic drinks are produced with distillation. some whiskey, brandy, rum and all that and all getting produced with this. Okay. Then without distillation, wine and beer and all without distillation. Both are alcoholic drinks. Alcoholic drinks may be formed with distillation and without distillation. With distillation, which and all whiskey, brandy, rum and all, and without distillation, wine and beer. Okay. Wine and beer, they are the products which are produced without distillation. Then the products which are produced by distillation of the fermented broth. That fermented broth is getting distilled. Then the product is getting formed. Okay. For that, examples are whiskey, brandy, rum. Okay. Here, these are the pictures of the. Here you can see the plant, fermentation plant, and these are the fermenters. Okay. Next, we are moving to antibiotics. So now we have learned about the role of microbes in the production of what? Production of alcoholic products. Then next we are going to learn about antibiotics. Okay. We are going to learn about the microbes in the production of antibiotics. So here, you know that is a very good contribution to the society. Right? To human society, it is a very good contribution. So the antibiotic discovery, it is a very significant discovery in 20th century. Antibiotic discovery in 20th century was a very good or very important or very significant discovery. So what is the meaning of that? That word antibiotic. What is the meaning of that word antibiotic? Anti means, here you can see antibiotic. What do you mean by anti? Again, something we are saying means that is anti. Against, right? So anti means against something. Antibiotic means it's related to life. Biotics. This word is related to life. So antibiotic means against life. Right? So antibiotic means against life. So antibiotics, they are the chemical substances which are produced by microbes. And these products that can kill the growth of other disease causing microorganism. Okay. So they can kill the other microorganisms growth, or they can reduce the growth of other disease causing microorganism. So what are antibiotics? Antibiotics means they are the chemical substances antibiotic. They are the chemical substances. Chemicals that can reduce the growth okay, they are the chemical substances which can kill or which can reduce the growth of other disease causing microorganism. So for example, we can say antibiotics, the most important example, that is penicillin. Okay. Penicillin is the best example for antibiotic. So it is commonly used antibiotic, this penicillin. And penicillin is the first discovered antibiotic. First antibiotic. Penicillin is the first discovered antibiotic. And who discovered penicillin? Alexander Fleming. So Alexander Fleming, he discovered penicillin, and the discovery of penicillin, that was accidental actually. See, actually Alexander Fleming, accidentally he discovered this antibiotic penicillin. So Alexander Fleming, he was working on Staphylococcus bacteria. Actually, his work was going on Staphylococcus bacteria. So he kept some culture plate, this bacterial culture plate, that Staphylococcus bacteria culture plate he kept, and they didn't wash actually. So in some unwashed culture plate, they saw some other growing mold. Okay, some another mold is growing, like that they saw, and they started observing that. So that other growing mold, around that this Staphylococcus that couldn't grow. Okay, so he found that that is due to the production of some kind of chemicals by that mold. And in the presence of that particular chemical, the Staphylococcus bacteria were not able to grow. So he tried to find out that, and he named that chemical penicillin after that mold, Penicillium mutatum. Okay. First they named that mold Penicillium mutatum, then he named the chemical penicillin. The release of this penicillin, due to that, the another staphylococci, they couldn't grow around it. Okay. So when he was working on the Staphylococcus bacteria, they kept some unwashed culture plate, and they were observing some other mold. Then around that other growing mold, that Staphylococci didn't grow, or they couldn't grow, and they found that that is due to the production of some particular chemical, some new chemicals by that mold. And that presence of that particular chemical, that Staphylococcus bacteria, they were not able to grow at all. So they named that, first they named that mold Penicillium mutatum, then named the chemical penicillin. Okay. Later, after some period later, Ernest Chain, another scientist, Ernest Chain and Howard Florey. Howard Florey. Ernest Chain and Howard Florey, they established its full potential and effective antibiotic with that penicillium. Okay, they established that full potential, then effective antibiotic. Okay, with this penicillin, they got the Nobel Prize in 1945. They got the Nobel Prize in 1945. 1945, they got the Nobel Prize for discovering this effective antibiotic. Which is that antibiotic? Penicillin. They got the Nobel Prize. So after this work, so many other antibiotics, they, not only those scientists, other people also purified from other microbes. Okay, antibiotics, it is from microbes only. Okay, so these antibiotics, they are helping to treat so many deadly diseases which are there: leprosy, plague, whooping cough and all. So that kills many people in the world, right? So this kind of antibiotics, they are helping, these antibiotics are helping to kill such diseases. Okay. So what do you mean by antibiotic? Anti means against. Biotic means that what is related to life, or life we can say. Antibiotic means against life. So what is antibiotic? Antibiotics, they are the chemical substances which are produced by microbes. Microbes are producing this kind of chemical substances, and that chemical substances can kill or they can reduce the growth of other disease-causing microorganisms. Okay, the antibiotics means, what do you mean by antibiotics? They are the chemical substances which can kill or which can reduce the growth of other microorganisms which are causing diseases. Okay, so the example is penicillin. Penicillin, it is a commonly used antibiotic, and penicillin is discovered by Alexander Fleming. During another experiment, he did this discovery, and what he was doing, he was conducting some staphylococcus studies on staphylococcus bacteria, and he kept some unwashed culture plates, and in that they observed some other mold is growing, but that mold, around that mold, some chemical will be getting, or around that mold, the staphylococcus was not growing. So they came to know that due to the presence of some chemical which is released from this mold of the mold of this particular new mold, that staphylococcus could not grow. Okay, and with the presence of that chemical, the staphylococcus bacteria were not able to grow at all. So they named that chemical as penicillin after naming that mold Penicillium notatum. First they gave the name Penicillium notatum for that particular mold, and the chemical which is released from that is called as penicillin, or they gave that name. Then later, Ernest Chain and Howard Florey, they got the Nobel Prize in 1945 for the effective antibiotic. The full potential and effective antibiotic, and they received the Nobel Prize in 1945 for this discovery of this antibiotic. And after this work, so many other scientists produced so many antibiotics from different microbes. Okay, and these antibiotics, actually they are helping to treat several diseases like plague, whooping cough, leprosy and all, that is treated with the help of these antibiotics. Got it? So is it clear? Okay, so hope it's clear up to this. So just go through this. If you have any doubt, you can ask me in the next class. Just learn about antibiotics. These are very important for NEET as well as your exam, normal exam, and for NEET also. This is very important topic. They'll ask about different types of antibiotics and all. Then the other things which you have learned is viroids, prions. These are also very important topics. Today, whatever we have discussed, all are very important. Then industrial products we have learned. So which are microbes which are produced used in the brewery, then bakery. That names, the scientific name you have to remember. Then another one, antibiotic, then different antibiotics. This and all we need to learn. Okay, thank you.